So my job here is to talk about your job before you plan a metabolomics experiment, how to plan for a good study design with metabolomics angle in mind. As mentioned earlier, when you have an idea or a hypothesis to test, come to us, consult with us. We'll talk about the study design and help you with the small, minute changes that make a huge difference in metabolomics experiments. We'll talk about how you can plan sample collection, storage, handling, and so on. So in my talk, I'm going to be talking about how to design a good metabolomic study, what are the effects of different sample collecting strategies on your results, how to store and handle these samples, and how to plan for data normalization before you actually do the analysis. So initially, we'll talk about the different study types. You guys already design experiments. This is not new, but I'm just going to put a metabolomics angle to it. How much sample size is required, the different model systems, the different biological materials that we can work on. So as with all clinical experiments, we need a well-matched control cohort for all metabolomics experiments. I cannot stress this very well enough because these are comparative platforms. There's no published reference standards for most of these by these techniques. Therefore, you need a well-matched control cohort. In interventional studies, these are very well suited for our purposes because here you can compare before and after the intervention. Therefore, reduces the differences between individual phenotypes. Crossover trials are even more ideal because here the placebo and the intervention is done to the same group. Therefore, we're essentially comparing only the intervention effects on these cohorts. A common question that arises is, what about the sample size? How big should my design be? The reality of it is these platforms usually measure a lot of variables. Therefore, the increasing variables forces the false discovery rate to be high. Therefore, the more the sample size, more the power, more the limiting the false discovery rate. Now I'm going to talk about the different model systems, cell, animal, and human. In cell studies, these are unique in a sense that this gives us a position to both study the cell intracellular metabolome or the endometabolome, or the exometabolome, that is the culture media that things are secreted and the cells interact with. We can also feed in isotope labeled traces into the cell culture media, which enter the cell, go through the different metabolic cycles that you're testing, and also test the positional isotopomers that are different according to the flux in the metabolic cycles of these cells. These are more reproducible model systems because you control everything in this case. You control what substrates you feed the cells, you control the temperature and everything. Therefore, very reproducible, small sample numbers are okay. Since you control everything, minor changes in your experimental condition make huge changes to the metabolic profile of cells. So the rate of metabolism is entirely under your control. The seeding densities of the cells that you start with matters because this leads cells to either go into proliferation or growth arrest, and that makes changes to your metabolic profile. The culture time, the passage number, the cell age, all that make a change. Growth conditions, temperature, ambient atmosphere, whether the carbon dioxide is present, amount of oxygen in the atmosphere that you grow these cells in make a difference. You also feed in the nutrients for these cells, the amount of glucose in your culture media, amount of glutamine, and how often you change your culture media, all that makes a difference. As an example, I have here a study done on myeloma cells where the amount of glucose present in the culture media is measured. This is the x-axis is the number of hours the culture was conducted in. And you can see the glucose concentration decreases pretty fast and the lactate concentration goes up. And if you're going to sample at this level here, you're going to have a very inconsistent results. Therefore, consistency is the key word here. Whatever you do, your experimental conditions need to be consistent and sample at the same time. Also, though you control all the metabolism of the cells that you're growing, how to capture that at that instant so that you can feed it into our analytical system. So how to capture metabolism, freeze it, metabolism quenching matters. For example, in our study in our core here, this is beta cells, clonal beta cells that were grown, and the glycolytic pathway was studied in a targeted manner. And this is the difference between quenching the metabolism between liquid nitrogen and solvents at different temperature, minus 80 and 0. As you can see, these asterisks show the differences this step is making on the metabolic profile that you get. Therefore, in this instant, we concluded that liquid nitrogen was the best way to stop metabolism. But if you do decide to choose any other option, be consistent throughout your study. Also, how are you getting the cells out? Are you going to scrape it out? Are you going to do enzyme digestion? Whatever you choose a consistent thing. So you do that through an entire experiment. Also, cell lysis happens when you centrifuge a cell pellet at high centrifugation speeds. So that needs to be considered as well. Animal studies are a little more easier because you control what mice you're going to use for your experiments. So they're generally from a similar genetic makeup. You control the ambient environment, you feed them the diet that you choose. So more reproducible inter-individual variations are minimal. Small sample numbers will do like 8 to 10 per condition that you want to test. Also with the animal studies, as you all know, when you sacrifice, you get tissue, blood, urine all at the same time. Therefore, you can compare the metabolic profiles between all these biological fluids. Therefore, kind of figuring out where the flux of these metabolites are happening. Also, in this case, I want to reiterate the need for consistency. For example, one of our core experiments, we had a group that did mice experiments. They started off with sacrificing the mice by cervical dislocation. But towards the end, they started adding an anesthetic to the end of life experiments. Of course, guess what the greatest peak of separation was? The separation was the anesthetic peak. Therefore, what you do matters. If you don't maintain consistency or don't let us know about the experimental changes that you made, those would be things that matter. These are things to be consistent, and you need to consult with us. We are available all the time for consultation, so talk to us before you decide the study. Human studies, because of the variables that we're going to measure, we probably need a big sample size. But one good strategy is to start with a small discovery cohort, 10 to 20 patients, and see what the effect size is. Because most often time you're testing a hypothesis that has not been tested before. Therefore, you have no clue about the effect size. And once you get an idea from the discovery cohort what the effect size is going to be, you can plan a validation cohort larger, powered well to confirm your findings. So what are the biological materials we can test? We can test a wide range of things. We can test plasma, serum, whole blood, urine, saliva, CSF, tissue, a million or not cells. If you give us, we can test that. Culture media, as I talked about, to test the exometabolome. More specific instructions about how to collect these samples are present in the website. This is the link. It's present in all your slides. We can work with a variety of sample volume, depending on what you want measured with your samples. We can work with as little as 50 microliter in case of plasma and serum. Sometimes you need as much as 5 ml for urine because you want to measure steroids in the urine, or something like that. Again, come to us, consult with us, and do a very consistent experiment. We'll work this together. People also question me about pooling, especially when the sample quantity is very low. You want to have as many technical and biological applicators as possible. It is possible, decreases cost and time, but obviously when you pool, you don't know what your outliers are, what are going to drag your differences to. You're also unable to detect after the fact which sample did what to your difference. Next, I'm going to talk about the timing of sample collection. I really want to emphasize this. This makes a huge difference, and I'm going to have illustrative examples about this. This is a study here. About 10 patients were made to not sleep for 40 hours, extended wakefulness. I'm talking about this study to illustrate the effects of circadian rhythm on your metabolites, especially the steroid aspect of things. Here, we are measuring dihydro palmitate and other steroids that we're measuring and fatty acids we're measuring here, you can see that as the number of hours go, there is a distinct pattern here. This is the 24-hour mark. You can see that despite not being going to bed, there is a distinct circadian rhythm. So what does this mean to us? What time you decide to choose to draw that sample matters. So be consistent with the time of collection of your samples. This slide here is from a study to illustrate the effects of fasting, feeding, physical activity, and stress on your metabolic profile. This is time in hours, and here we are measuring two metabolites. One is free carnitine, and the green one is the combination of all different acylcarnitines circulating. This is plasma in 15 subjects. As you can see, as the time in fasting increases, the profile changes. You feed them a liquid diet, changes again. This is a liquid diet again. The glucose tolerance, the pattern changes again. This is physical activity, which is 1 hour on a stationary bike. You see the difference again in the carnitine profile. This is a lipid challenge. You can see again the differences in the carnitine profile. This is stress. In this case, it was cold stress. They made them put their hand in cold water for 3 minutes. That also makes a difference. So what I want you guys to take from this slide is that where your patient is or subject is in this fast feed physical activity cycle also matters. So the standardized sample collection at a time of day in relation to meals and physical activity. This slide here is from a study where persons were fed 10 healthy subjects were fed standardized diet for 10 days, and then they were brought into the lab and fed a breakfast basically full of saturated fatty acid, monounsaturated fatty acid, or polyunsaturated fatty acid. And then they collected the urine at 2, 4, 8 hours, and this is a PCA plot of the differences the diet had made in the urine. As you see, the diet, one single meal makes a huge difference in the urinary profile. So what does this mean for you is standardize the patient's diet, or if that's not possible, at least maintain careful diet history so you can put that factor in it to your analysis. So the strategies to prevent variability is time your samples to collection to a certain part of the day to take into account the circadian rhythm. Time it in relation to physical activity, meals, also standardize the diet, if not at least get a good diet history. Careful medication history, I haven't mentioned it here, but obviously in a diabetic, if you give insulin, the profile changes. The lipid, the amino acid, and the glucose profile changes. Next, I'm going to talk about sample collecting strategies. So we do this routinely in our lab. We choose a specific collection tube, we choose a specific collection method, we store it in a collecting temperature. This matters a lot in metabolomics because these metabolic profiles are very sensitive. So we'll talk about this briefly. So this is the different sample collection tubes make on blood collection, whether it's serum or plasma. In this experiment, they took blood from 10 healthy subjects, pooled it together, made a pooled plasma, and they collected it in different tubes, EDTA containing tubes, citrate containing tubes, fluoride containing tubes, lithium heparinate tubes, and collected serum with nothing in it. Simultaneously, they made a solution of albumin and collected them, called it the blank, and collected it in the same tubes. And you see the spectrum in these different tubes, and you can see the chemical noise or the artifacts are highest in the lithium heparinate tubes. So the tube choice makes a difference. We think here in our code that EDTA plasma is the best, but we can work with you. If you have serum stored from a previous study, we can work with you. But as long as the entire experiment is serum collected in a specific pattern, we can work with you. This is the effect of hemolysis on the metabolic profile. Blood collection always produces some amount of hemolysis. In this experiment, 10 patients, blood was collected from them, and blood was collected from them and put through a PCA analysis. You can see the different hemolysis strengths separate out differently and show a lot of inconsistency. The most consistently correlating one is lysophosphatidyl that goes along with the free hemoglobin in the plasma. Therefore, the best thing would be to avoid sample hemolysis or at least eliminate those samples with hemolysis for our analysis. This is the effect of bacterial growth. This is most relevant in the case of urine. If you set out urine in the lab bench and not stored properly, this could happen. This is an example of a single urine. This is a normally stored urine. You can see the spectrogram here. This is a creatinine peak. This is the same urine left on the bench. You can see new peaks develop here, here. This is the spectrogram of the difference between these two samples, and the peaks identified by TMAO, which is trimethylamine oxide that Dr. Penetho mentioned earlier, and also the level of acetate increases. So how to prevent this? You can centrifuge, remove the sediment from urine. You can also filter the urine through bacterial filters. Also, addition of sodium azide to urine before storage prevents the bacterial contamination. Next, we're going to talk about how to store the samples that you collected so efficiently. Now we're going to talk about the effects of room temperature. So this study, 10 samples with plasma collected and left out for different hours, 2, 4, 8, 24. We're just focusing on two metabolites here, hypoxanthine and sphingosine 1-phosphate. You can see the levels keep increasing as the exposure to room temperature increases. So this is obviously an issue. So the strategy to prevent this would be to work on ice while processing the samples, limit processing time. Also, as soon as possible, I know sometimes there's limitations, but as soon as possible, store it at -80. And when you transport samples, please transport them on dry ice to maintain the -80 temperature. This study here talks about the effects of freeze-thaw cycles. Sometimes we store samples in large volumes and we take out aliquots many times from the same sample. What does that do to our metabolic profile? This is just a measure of carnitine in such one pooled plasma sample. You can see in the fresh sample, this is the level of carnitine. 1 freeze-thaw cycle, 2 freeze-thaw cycles, 4 freeze-thaw cycles. And you can see the carnitine difference becomes significant. So you need to limit the amount of freeze-thaw cycles. Or if not, at least be consistent throughout your experiment about how many freeze-thaw cycles you're going to have. Okay. Now we're going to talk about data pre-normalization, because sometimes we do the experiment, everything is done, and you go, how do we normalize this? So a little bit mention about this. Obviously, with cell experiment, we can do cell numbers. With liquids like plasma, serum, urine, you can use volume. With plasma, you can use key metabolites like glucose and creatinine to do the normalization. With urine, specifically, we nephrologists use creatinine and osmolality for normalization. If you have other biological fluids, protein concentration works really well, especially in tissue that you've homogenized. You can also use dry weight of the tissue, if that's available. And if you have forgotten about all these steps, you've run the analysis, don't worry, we can still normalize with the total ion chromatogram that we see in the machine. One other note about things, sometimes customers, they send different batches of the same experiment to run at different times. There is, as Dr. Penetho talked earlier, there's significant batch effects with each analysis. The best way to do things is to collect all your samples, store them at -80, finish your experiment, then run it in one run to the mass spec. That limits the batch-to-batch differences and keeps consistency. So in conclusion, a good metabolomics experiment needs to have a carefully thought of, standardized operating protocol. You need to think about how you're going to collect the sample, what time you're going to collect the sample, how you're going to store the sample, and how you're going to analyze the samples by normalization. And if you took anything at all from my talk, remember these two words: consistency and consultation. And these are the references I alluded to in my talk. And I can take questions.
Have you looked at sodium versus lithium heparin for plasma preservation?
Heparin by itself gives rise to a lot of artifacts in the sample because the way the polymers of heparin split out. So I don't think sodium or lithium would make a big difference.
There's some evidence that there may be higher endotoxin levels in lithium heparin versus sodium heparin. And just in the clinic, the green top tubes are mostly used are sodium heparin. So I just wondered if you...
Heparin itself makes a huge skew. So regardless of sodium or lithium, compared to EDTA plasma that we see, there is a huge difference in the chemical noise. But if you have consistently the same tube used for your study, we can still work with that.
But it is well known that lithium actually attacks cell membrane, so lithium can be worse.
Yeah, I mean, there's other, lithium heparin versus sodium heparin itself.
Other issues.
What it might do to the sample.
Yeah.
Could.
But in general, what you want to do is stay away from heparin.
Yeah.
Right.
Because all you can. I mean, that's the.
If it's not possible for your measurement reasons, if you consistently do it, the same lithium heparin throughout, we can measure it.
So in terms of normalization with glucose, I'm not asking a whole lot of detail, but do you mean you would measure glucose in a patient sample at the same time as you're collecting your sample that you'll later analyze?
We could do that, or you can do it in the routine fashion with the AccuCheck or regular chemical lab as well. We could do that with our technique as well.
So tissue culture cells, it's best that, a lot of times what we suggest people do is if you can grow another set of cells under the conditions that you can analyze, you can have a separate protein assay that's going to be when you harvest the cells. Because you can do protein on the precipitate, but it's not as consistent as having our separate cell. We're trying to develop some techniques to perhaps use a membrane of lipids as an internal normalizer. Because phosphatidylcholine is some of those lipids are very consistent between cells, but those can be changed by different metabolic conditions as well. And so, I think the last two words that were on the slides is consistency and talk to us.